Unit 2, Essay Question 3. So we're going to talk about the muscle sensory receptors. We'll start first by kind of defining where they come from and how they get to certain areas of the body. So we'll just draw the spinal cord. We'll call this a little nerve coming out. So the major alpha motor neuron will come out and it'll branch out to a couple of different places to some muscle fiber cells. The fourth one down here. The gamma motor neuron, which I'll show here in red, is going out and following these things. So here, to there, to there. And then the sensory muscle spindle apparatus is going backwards. So red is moving in this direction. And then these blue ones that we'll draw are going to move back towards the spinal cord. So they'll converge and head back in. And blue is going to head in that direction toward the spinal cord. So these sensory muscle spindle apparatus cells, if we blow one of those up like this, then inside there are two types of fibers. So you'll have these, which are the nuclear chain fibers, and then you'll have another type of fiber called the nuclear bag fiber, and that's kind of exactly what it sounds like, where it's a bulge. That is a stretch receptor. So the nuclear bag fiber would be, let me draw one more kind of sideways here. And then we'll draw one more on top. We'll talk about this in a second. So this will be innervated here. Excuse me. This will be innervated by a dynamic gamma motor neuron. So dynamic gamma neuron. And then this one by just the gamma neuron, motor neuron. So there are receptors that stretch along this bag of fiber that are in association with the 1A type of fast twitch muscle, whereas these are having to do with type 2 muscle fibers. So this gives rate information as the muscle is stretched for how long, for how much, and then this is for how long. It gives the duration information. And it's just got little fibers in there that are receptive to these things. And then we have, after we have these, it sends the signals back into our spinal cord. So let me draw kind of what a spinal cord looks like. Just 1 second. Okay. So we're going to draw in blue. We're going to have one of these sensory motor neurons coming in this dorsal root. I'll have a synapse here, the release of all that acetylcholine, and it will come to two places. So we have a synapse here and a synapse here, both headed back out of this root. This is our alpha motor, and this is going to be the antagonist. So as this is coming in, we will have it move all the way to its alpha motor neuron. And because of this, we actually have a secondary branch that moves out and will synapse here, and it creates an EPSP, and we'll synapse on that. And then this small neuron here, this is called an interneuron. And that interneuron will synapse here. So this one was an EPSP, and this will be an IPSP synapse. And that will cause the antagonist muscle to not work, which is good because it will rip your tendon, it'll rip your bone right off. It'll cause a lot of damage to you, which is not good. And then if you have such power in your body, or we do have such power in our body to cause damage to ourselves, and so we have what's called the Golgi fibers. And so what these will do is they come in as well, but there is an interneuron here, so it will synapse there. Oh, excuse me, oops. It will synapse right here, so release some neurotransmitters and synapse there, causing the IPSP there and the EPSP here. But its main synapse will go to the antagonist muscle, and its goal is to totally shut down and stop that muscle from basically ripping the bone apart in your own body. It's there to protect you. These spindle fibers so these spindle fibers that we drew right here, they always keep tension. They are what will help us to know what kind of reflex is needed, and whether there's ways to test if there's been damage or not to those reflexes. The stretch and withdraw reflexes can be talked about using this setup here. So when we have receptors in our body that tell us when something is painful and we need to withdraw from those things. So say you touch a hot stove, and so you'll have a lower motor neuron go to your spinal cord. Spinal cord will tell you that you need to pull away, and so you'll have this sense sent into your spinal cord here, and it synapses straight to the alpha motor neuron, and we get this that fires, and it tells our flexor muscles in our arm to pull away, and at the same time the antagonistic muscles are being told, don't do anything, let us pull away from this this pain. And the same can be done with the stretch receptors. When a muscle is stretched, say the hammer reflex, which is very common, when the hammer hits your knee, it sends this signal in that your muscle was stretched and that the alpha motor neuron here needs to react, and so it shortens quickly and it flexes that. When it comes to taste, we use our tongues to taste. The surface of our tongue is pretty rough and kind of ridged and stuff like that, and it kind of looks like this. And in those ridges off of these, there are cells that are our actual taste buds, and there are approximately 125 cells per rough area like that. The general shape and kind of function of these cells looks like this. We'll draw them in a box shaped and they're kind of split into almost halves, quite close to a third. There's an approximate split here where there's vesicles full of neurotransmitters down here, full of all these different neurotransmitters. And there is a synaptic cleft here with a synapse in it. There is a voltage-gated calcium channel here. And there is a sarcoplasmic reticulum here, full of even more calcium. None of the physiology in this taste bud will depolarize and actually depolarize past threshold. It'll constantly give us graded potentials where we get close, but we're never going to reach it. And those 5 different things that we can taste are salt, sweet things, sour things, bitter things, and then finally umami things. So the way we taste salt is through ions. Specifically for this, we'll talk about the sodium ions. We taste we taste sweet things through carbs. Sour things come from acids, so protons or hydrogen. And then bitter are quinones. And then umami is through glutamate, the neurotransmitter. So now we can talk about how these go through these taste cells and how we actually are able to perceive them as taste. So let me draw 3 more boxes here, and we'll talk about each one of them and how they're set up. We taste salt a little bit differently than everything else. We taste sour a little bit differently, and then we taste sweet, bitter, and umami approximately the same way. So we'll do a salt taste cell first. In the salt taste cell, there is a channel here, and they are called ENaC channels. And this will allow sodium to come into the cell. And like I said before, there's a sarcoplasmic reticulum and a bunch of vesicles of neurotransmitters already down here with a voltage-gated calcium channel here. So as the sodium will move into the cell, it will affect this calcium channel to open, and the calcium will rush into these and grab the snare proteins, and it will move these neurotransmitters out. And those neurotransmitters allow us to taste the salty things that we eat. For a sour taste cell, again, we've got that sarcoplasmic reticulum and some vesicles ready to be released. We have the same situation where we have an ENaC channel up here. Let me write that, ENaC channels. And that ENaC channel for sour will let hydrogen in. And just like in most cells, there are potassium leak channels here. And this leak channel will actually be shut by the presence of this hydrogen. But there is still the calcium channels down here. And that calcium channel will, again, open because we're not letting these positive charges leak out through the potassium leak channels. It will, again, cause the snare proteins to push out these neurotransmitters, and it will let us taste sour things. And then finally, for sweet, umami, and bitter, it is slightly different. Here we're actually going to have a little G protein with a couple little subunits on it, and a phospholipase C. So phospholipase C, when these subunits are introduced to either the quinones, the glutamate, or the carbs, they will release IP3 into this cell. IP3 is the precursor for calcium, and so lots of calcium is going to be dumped into this cell very quickly. And so along with the calcium that is in the sarcoplasmic reticulum, these neurotransmitters get dumped, and you will taste the sweet, bitter, or umami flavors in your foods. Okay, so I will quickly talk about the sense of smell. So I've drawn, this is a nose. When we zoom in, this is the bone that separates your mucous membrane from the olfactory bulb. The olfactory bulb is where your sense of smell will be directed to your brain. So you have a synapse here that is heading into your brain, and there's receptors that come down into the mucous membrane, which is where you pick up those chemical receptors. So it goes through the bone and into the olfactory nerve. So if we blow this up, it looks like kind of a dendritic opening. And on this opening, there is a G protein that is coupled to a receptor known as adenylyl cyclase, which we'll put right here. This is adenylyl cyclase. Adenylyl cyclase. And when a sense hits this G protein, adenylyl cyclase is activated and it releases CAMP. When CAMP is released, here on the length of this, there is a couple of different channels. One will open up to let sodium in. One will open up to let calcium in. And then there is also a channel up here that actually it's a chloride channel, and the chloride will actually be released as the sodium and calcium enter into the neuron. It will start to depolarize, and as the calcium leaves, it depolarizes further, and that's where we get our action potential for smell. And that is how we perceive smell. The last thing we'll talk about for this question is the sense of hearing. So I drew some of the ear. This is the outer ear. This from here to here is the inner ear, and then this is the cochlea this is the middle ear, and this is the cochlea of the inner ear. This red line that I've drawn is the basilar membrane. When a sound hits our ear here and it starts to propagate sound waves inward, it reaches the tympanic membrane, which is called our eardrum, which sits right there, and it starts to vibrate the three parts of the inner ear. But the way we actually hear is if we take, this is the cross section, this is what this looks like if we cut this in half there and open it up. So we have a few different bodies here. We have the perilymph, which is this section, and the endolymph here, and this is also endolymph down here. And then this outline is the tectorial membrane, which is where we have these hair cells that attach. The hair cells attach into that tectorial membrane, and these little protrusions of the hairs, they are what cause us to hear. So if we blow one of these up, this is all the tectorial membrane and part of the endolymph. And then we'd blow this up here. So each one of the finger-like projections here contains a mechanically gated potassium channel. Inside of this endolymph, we have 150 millimolar per liter of potassium. And out here on the outside, we only have 5 millimolar per liter of potassium. And so following its gradient, it wants to leave the cell. The way this works is when vibration hits it. I also realized I drew this wrong. That calcium channel goes here. When vibration hits it, this mechanically gated channel will swing open, and therefore the channel here that's attached swings open, and the channel here that's attached swings open. Now, because there is such a high tendency of the potassium on the inside, there are positive cells all through here. It's charged in the positive direction, and then on the outside it's negatively charged all the way across the membrane. When these mechanically gated potassium channels open because of the vibrations, this potassium will rush into this cell. As it rushes in, it will, because what wants to happen is these positives want to meet these negatives to cancel. And so as the potassium rushes into this cell, we have the sodium potassium pump already working here, which is this. And so there's potassium already coming into the cell, and at that same time there's another leak channel for potassium here. So as it is able to reach into here, and it goes through the mechanically gated channel, and it finds the leak channel, and it comes out, the calcium channel, which I drew on this one, which would also be here, starts to open, and it releases calcium into the cell, which induces exocytosis by grabbing snare proteins that grab these vesicles, which I've drawn here, that will move the neurotransmitter to the plasma membrane, and it will release out into the synaptic cleft here, and it will send action potentials down this neuron, and that's what allows our brain to perceive what we are hearing.